Today, I'm going to give a brief overview of how sampling technique, a seemingly inconsequential study variable, may have major downstream effects on experimental outcomes in studies using non-human primates. As a quick background, primates are often used as models to evaluate the safety and efficacy of novel therapeutics and their suitability for the clinical setting. This includes the evaluation of new drugs, new transplant protocols, and new vaccines. To maximize the translational value of these studies, it's important that we reduce any extra influences on the model that may interfere with our evaluation of the novel intervention and that would not typically be seen in the true clinical setting. This leads us to a discussion of the possible differences between restraint-based and cooperative sampling and why these differences matter. In preclinical studies, primates often undergo frequent handling and sampling to assess the effects of a novel therapy and how these effects change over time. This includes the evaluation of safety indices such as CBC chemistry profile changes and the performance of physical exams to check for adverse reactions, in addition to the evaluation of efficacy, including multiple aspects of the immune response. This sampling usually occurs relatively frequently, such as weekly or more in the example here, and may potentially introduce a source of stress to the animals. The most commonly used method for sampling involves restraint, often in the form of sedation. While sedation makes sampling and performing a thorough physical exam fairly easy, it has the potential to introduce both acute and chronic stress, along with possible confounding influences from the sedative chemical itself. It should also be noted that this is not representative of how sampling usually occurs in the clinical setting, which may further impair translatability. By comparison, a less commonly used approach involves cooperative sampling, made possible with positive reinforcement-based training. In this method, animals develop positive associations with handler interaction and are made more comfortable with routine medical procedures. This should help reduce the introduction of stress during sampling and is more representative of what actually occurs in the clinic. To show why stress matters, we provide a brief reminder of some of the physiological effects it may have in relation to some of our important outcome measures. In this model, we propose that repeated sedation activates the stress response in the HPA axis. This has a long list of effects, including changes in vitals and immune functioning. Effects on the immune system include changes in antibody production, cytokine expression, immune cell differentiation, and activation of the inflammatory response. All of these changes can have major consequences for the interpretation of study results, especially in studies that are trying to evaluate the immune response to a novel therapy. To determine whether these effects actually differentially exist based on sampling technique, we have begun to investigate whether differences actually manifest in some of our outcome measures. In the example shown here, we compared average serum cytokine expression between groups of cynomolgus macaques that were sampled either cooperatively or while ketamine sedated. Overall, we found a widespread blunting of cytokine expression in the sedated group compared to the cooperative group across a wide range of cytokines, including those that are known to have major pro-inflammatory roles and those that are known to have major anti-inflammatory roles. This shows that, regardless of precise mechanism, it is likely that sampling technique has an effect on baseline immunology that may have profound impact on study interpretation. Overall, based on the known effects that stress may have across physiological systems, and based on our initial findings on the effects of sampling technique on cytokine expression, careful consideration must be made in how to perform sampling in preclinical trials. Refined sampling will help produce results that are most relevant to the clinical situation and help reduce the effects of confounding factors. Overall, this will improve the translational validity of many of these preclinical studies and will produce results that are most relevant to the clinical setting.